Good evening, students. Most welcome to everyone for today's third session of animal cell culture. Um, Hope you can see my screen and everything is being shared nicely. And we are good to go for our workshop of today. Hope in your life also everything is going well, everything suits well. And thank you very much for your nice presentations. I really like some of them, but this one I don't like it. Not so good. It's just done to make it done for the sake of there's signature of also. Some are really Arun Kumar. Hmm, okay. And some were really nice, very well ordered. Like this one is good. Like you made some pictures, some light microscope, electron microscope, SAM, TAM. And I hope you also like today's MCQs. So try to increase your Srijani, to increase your pixel size. From here you can increase your pixel and then you can check it. Thank you for replying on the MCQs today. So I got some MCQs in the morning, so I just feel like sharing with you. And most of the you are replied really nicely, and I hope you also have your freshen up your knowledge. So I will keep sharing these MCQs for related to some topics that we do on the next day, so that we are able to understand how things are working out. So let's start with this. So only 56 students have been joined so far, and there are around 170 students. So this is not fair that you had paid for your workshop and you don't just you're coming for the sake of certificate. Believe me, it will be of no use if the students are coming for the sake of certificates. How many were present? 52. So we haven't even started the advanced topics yet. There's a thing called attendance actually. If you are all gonna be so less present, it will be not uh, good. Okay, let's start. I hope students will join now after seeing this message. So today we will start with animal cell culture. How many of you have done this thing in your life with animal cell culture? Uh, yeah, that's a good idea, but let me see for a couple of minutes. From the tomorrow onwards, we can shift because we are told that it has, because I cannot record the sessions. So to record the sessions, I have to be on YouTube. We cannot record on the Google Meet. So who have done animal cell culture? Let's concentrate on the work now. Let's forget about which platform should we choose. Sci students are not coming. Let's start the studies now. Yeah. Okay. Somdatta have done that. Bhavika have done that. Okay, that's good. So animal cell culture. Okay. Okay. In class, Neha Taneja have done it. Oh, that's good, Kanwal. So today I will be giving you a little brief about this, how this is done, and good, 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 good. Let's start. So basically, cell culture in vitro, it's a brief history. In 1885, first of all, your rocks maintain the embryonic check cells alive in saline solutions for a short length of time. Then in 1912, Alexis Carrel cultured connective tissues and showed heart muscles tissue contractility over 2 to 3 months. In 43, Earle et al. Produced continuous red cell line. In 62, Bonassi they published for maintaining differentiated cells. And in 70s, growth media, growth factors for many cell types were designed. And in 79, Bottenstein and Sato defined serum-free media for neural cells. In the 1980 to till now, tissue culture became less of an experimental research and more of widely accepted research tool. So, what are the application of cell culture techniques? It helps in the recombinant DNA technology, drug effect, vaccine development, artificial organ development, characteristics of the cells to maintain over the long period of generations. Can be cells can be exposed to various reagents such as radiochemicals, drugs at different concentrations. Yeah, and there are some limitations. We have to develop standardized techniques. It takes some time to learn these aseptic techniques because you have to create everything clean with your 70% ethanol. Quantity of material is limited, and the cost of medium maintenance of cell line is also quite high. Just a second, students. I think. Wait, wait, wait. I think this is not the presentation I have to teach you. Just a second. I have to teach you one. I think this is not the one I have to teach you. This one. So in tissue culture laboratory, there is some fuzzy disciplines that you have to be on bench top. It has to be very keep it clean and clear so that no bacterial contamination should be there. And we have to wear long sleeves lab coats because even the hairs on our body should not interfere with our cell culture. Even a small droplet of hair from our nails or anything could go into our cell culture. It could contain a very biggest mistakes or life. So everything has to be taken care. um, Wear gloves while doing a tissue culture, which minimizes contamination from skin organisms. Surfaces, gloves, solutions, plasticware sprayed with 70% alcohol before placed in the biological hood. Solutions, reagents, and glassware used in tissue culture work should not be shared with non-tissue culture work. So keep things that other things are not coming up into your tissue culture would work. Then what are the primary applications of animal cell culture? We can maintain the cell cycle control characteristics of cancer cells, detection and production of functions of growth factors, study of various specialized cell functions, cell and cell matrix interactions. So there are various types of cell culture. There's a primary cell culture and cell line. What is primary cell culture? It's basically is a fresh isolated from tissue source. The cell line, finite cell lines dies after several subcultures, and continuous cell lines transferred immortal. So I have done in my life cell culture with the cardiomyocytes here. It's also shown that the myoblast is being done with the YAFE, and we can store them in liquid nitrogen for longer time, in quite long time, at minus 70 degrees Celsius. So what is passaging or subculture? So once you have a culture, you cannot have that for a long time. What will happen? It will start producing primary metabolites, secondary metabolites, and with the time, things will be really bad for the cell culture. So what we have to do is then we have to, with the time, passaging our culture or subculture them, so that things could be like they could start having fresh medium. In passaging, we just give them fresh medium and divide the cells into two different parts. So here, the cells are dissociated from the flask using enzymes, and we slip it into two different flasks. There's a thing called contact inhibition. So contact inhibited monolayer of normal cells, and in this way they will be growing very well. But once they are overgrowth, so multi-layer inhibited cancer cells here, your cancer cell will start growing on. So you will see some piles of cells on the top, and they will even might coming out of your growth medium, which is not good for cell culture. So this could also happen. So how to initiate, establish, and propagate your cell culture? So cell culture can be initiated from tissue or organ fragments, or single cell suspensions. Choices has to be made. Disaggregation techniques, media, culture condition, selection procedure has to be taken care. Then following things has to be considered while making your cell culture. Sensitive to mechanical dispersal enzymes or cell-cell contact may be required for proliferation in this case. Dispersed cells in cell cultures are vulnerable. Mostly primary cells will be satisfactory and adhered. Some cells will be prone to spontaneous transformations. So limited lifespan of some cultures could be seen there. And how you can disperse your tissues? You can do it with the mechanical method by mincing, sharing, or sieving, or chemical or enzymatic method. So once you have a cell culture, and then you have to passage it or split it into two different flasks, or growth into a different medium or enzyme. So you have to remove it from the bottom of your cell culture plates. So you can do it with the help of mechanical method, or chemical, or enzymatic. In the enzymatic methods, you can do it with the help of trypsin, thronase, collagenase, dispase, and or we can combine all the above methods. So cell culture environment. So this is 8-well cell culture dish. So allows comparisons of 8 samples, can have 8 different stains, fixed at different times. Then remove wells and gaskets, Leave one slide with 8 separate samples for easy microscopic analysis of cells. Then you have 96 well plate, which allows comparisons of culture conditions. Samples often triplicate. So on that note, but in 70% ethanol, so we use 30% water and 70% ethanol, so that the mixture that is present is quite appropriate to kill any bacterial infections. Otherwise, if we use 100% ethanol, it's not good for our skin also. It will start deteriorating our skin using 100% ethanol mostly. So that's why we keep some water. So this is the best quantity to go for. Yeah, higher percentage of ethanol will cause excessive dehydration over the required amount. But your phenol red, because this is the best. Neha, you have a good question. Whenever there is acidic conditions, your cell culture will turn into yellow color. Or whenever it's more basic, your cell culture will be more in pinkish color. And whenever it's in basic, it will be in red color. So that's why we use color in our medium. So it will help us to see how our cell culture is flourishing. If it is acidic, it will go to yellow color. That's why we have phenol red as a pH indicator. If it is more basic, then it goes to pinkish color. I did some experiment with heat shock proteins and some ER stress regulators. So whenever I was adding them into my cell culture, what I could see that the color was changing into yellow color. They were getting more acidic. Or when I'm using H2O2, which is an oxidant, antioxidant, so things were happening very rapidly different. The colors were changing quite drastically into the of my cell culture. But yeah, bromophenol blue will be toxic actually. So far I remember, I never use this, and it will be toxic for the cell culture. It will be not, I think, a good way to go for. Yeah, we go for passaging to stop this inhibition of cell culture. We will discuss which culture do we use. We will discuss about fetus serum medium and non-fetus fetus medium. We will discuss about this. So I want to show you uh, so this was my master thesis presentation. I given this in 2012. So see how a presentation has to be done. I wanted that I could give you some lectures over 1 week. We can have a workshop over this. How to give nice presentation, how to give confidently, and everything has to be in order with a very stagnant story. So it was done at the group of Dr. Stephen Offermans in Bad Nauheim, Germany. So my supervisor at the Max Planck at that time was Dr. Boris Trelech. So I worked on the novel endothelial specific molecules involved in the tumor cell extravasation. So it was basically I was checking at the metastasis thing. So here what we can see, we have a primary tumor which is invading into our transendothelial membrane. Then after once it goes into the membrane, it goes outside the membrane and then start to invade in the secondary tumor. So transport through the blood or lymph vessels. So this is how from the primary tumor it becomes a secondary tumor with the help of a blood, and then getting out from to the secondary part and then becoming a secondary tumor. If it is in the liver, then from liver it goes to your intestine or any other organ. So this is how your metastasis is done. So my task was to check with the row GTPases. I will not discuss about them. So this was my siRNA plate. So I want to show you in the 96 well plate that I was showing you in my cell culture. So here the 96 well plate. So each cell plate has within them a microporous membrane of 8 micrometer, and the lower chamber is empty. And what I used to do was I used to make a layer of Huvix cell line there, and on the top I put EGM media, which is required for the good growth of Huvix, and upper chamber was empty. Then I knocked down these cells with the siRNAs. We remove one cell from each of them. Then afterwards, what we used to do is we used to put cancer cells on top and let them transmigrate for 5 hours. And then we check at the bottom how many cells have been transmigrated. So we did this for all the 96 well plates. So each well will be have a different knockdown gene. So that's why we have a siRNA library. So there are around 256 siRNAs of Rho GTPases. So we choose all of those siRNAs and we start to knock down my Huvix cells, which is a human umbilical endovascular endothelial cells. So we used to get the knock down them, and then we transmigrate on the top with the cancer cells, and then check how many cells have transmigrated out of it. So this is how it looks like in the normal case when the Huvix were put into this 96 well plate. So then we check them with the help of microscope. So here I use inverted microscope, the automatic one, so which take image of each cell automatically and goes to the next. So it's a very highly advanced technique was available at that time. And then it was taking this image, then this image, then this image, and like this it collected all the images. Then I used the software ImageJ and I collected all these wells and made this 96 well plate wall. So what I saw at that time was that the 96 well plate, all the cells at the outer cell membrane, they were not growing very well. Only the cells which were in the middle somewhere, they were giving me a mono layer. So it was no distortion or not excessive growth of cells. Everything was fine. So it's maybe the outer side was not growing because of the being touched to the hands and not being available to the air mostly, and the inside membranes were all protected. Voice is too slow. I don't know. So let me continue now. So this was the first experiment was done to do the optimization of Huvec monolayer. Then after the optimization was done, I choose only the 96 wells inside well plate, and I also knock down with them control siRNA, which doesn't actually knock down any others, but mainly the um, normal ones. Then what I did, so I made some controls. So whenever you're doing experiments, remember to have multiple inner controls, that is your positive control and negative control. So here I choose my positive and negative control. So one was the normal control siRNA, which doesn't affect our siRNAs. It was just added to see that there is not any negative effect of it. Then CDHF5, these are the ones which maintain the good cell connection between each other. Then there's a killer RNA which kills siRNA, and then I also added calcine AM, which is a fluorescent dye. We can see in the microscope when I'm done with the experiments that everything is fine. I am seeing a monolayer there, so that's why I keep these two ones in between. And I did always them in duplicate. So this is RAS GRF1 or RAL GDS, RAP GAF4. So all these siRNAs were used in duplicates in one plate. On the other side, I also have these two different, the controls are being used in that case. So this is how I maintain my experiment. And then with the different SIRNs, this is how the your HUVEC cell line looks like in this 96 well plate. Your small cells being all together, and from these cells, the cancer cells are being transmigrated. Then this was the results that I found with the controls one, with the control CDH5 NK. This is how the Huvix monolayer looks like, and this is how the transmigrated tumor cells. So once the cells are being broken, so you can see some gaps, the more migration of cancerous cells are possible. In the natural killer cells, mostly your cells are died, then you see more transmigrated tumor cells out of it. So this is how you check the things for your experiments. So this was a control experiments. Then some remove some outliers, and this is how the image looks like. The transmigrated tumor cells from our HUVEC cells after the knocking down of the siRNAs. So this experiment, you should not be like, you need to understand everything, but it was just shown to you that how 96 well plates were being played used. Then I found some targets which were expressed, which were not expressed, and then based on that we got some results. Yeah, so I am not allowed to share with you any results here, sorry to that. Voice is too low, it's not possible. Please have some students, uh, please have some earphones towards to you. Some headphones so that voice could be fine. Otherwise, just check it later in the recorded version. I hope it will be fine. It sometimes. So, okay, students. So my main reason to show you my experiments in the 96 well plate was to show you how we can do the applications of animal cell culture with the HUVEC cell line or HeLa cells, COK cells, any cell line you want to use, stem cells you can use, cardiomyocytes, any cell line. Uh, so everything could be used to do your various experiments actually. So your yeast cells or any other cell culture. So that was the main reason to show you the applications of these 96 well plate experiments. So what are the factors that are affecting the cell behavior in the complex in vivo environment? So local environment, metabolites, local growth factors, your ECM architecture, everything plays big role. How the cells are interacting with each other, how the circulating proteins, cytokines, hormones being playing role there. So actually, in the cell culture, what we are doing is we are mimicking the in vitro what is happening uh, in body, and we try to mimic them in this artificial system, and we are trying to get the results artificially also by just using one cell line. But we have so many cells and so many enzymes, so many reactions are happening within one organ, so it will be not justified to say that whatever the happening in the cell culture will be happening in the animal cell culture or in the human body. So that's why first we do some experiments in one cell line, then we start doing experiments in the mouse to check at the bigger level, and then we can say that effect of our experiment, how things are working out. Then culture surface. So most adherent cell culture required to proliferate. We change charge of the surface and the coat with matrix proteins from like collagen, laminin, gelatin, fibronectin. So we can, so before growing our cell culture, So we need to make some a very nice surface layer to grow our cell culture, and for that we use all above things. Then media formulations. So initial studies use body fluids like plasma, lymph, serum, tissue extracts. And the early basal media, salts, amino acids, sugars, vitamins supplemented with serum. Then more defined media, we use cell specific, extremely complex plus serum in that. So this is your DMEM media, which has, here we can see, amino acids, yeah, vitamins, inorganic salts, and other components. So this is how the ingredients looks like. So in the amino acids, you have most of the important your amino acids: valine, tryptophan, threonine, serine, phenylalanine, methionine, lysine, leucine, isoleucine, cysteine, arginine, alanine, glutamine. In the vitamins, you have folic acids, choline chloride, inositol, niacinamide, yeah, riboflavin, thiamine, hydrochloride. On the other side, in the inorganic salts, you have calcium chloride, yeah, ferric nitrate, magnesium sulfate, potassium chloride, sodium bicarbonate, sodium chloride, sodium phosphate monobasics, other components, glucose, phenol red. So all these parts play a big role in making your media. And also there's inorganic ions which maintains the osmotic balance. Then there are some trace elements like cofactors for biochemical pathways, amino acids, vitamins, and energy source, as we have discussed just now. So there is also serum, which provides the following. There's a horse serum that we use mostly. It's also called as fetal calf serum, or we can use chick embryo extracts. So they provide the basic nutrients, hormones, and growth factors. Binding proteins like albumin, vitronectin, hormones, vitamins, minerals, protease inhibitors, pH buffer, all being used here. So these are the major serums components of the fetal calf serum. So this is how your short form is FCS. So we used to put this FCS. So what we used to do is we used to freeze them at minus um, at minus 10 degrees Celsius, and whenever we have to use them we again defreeze them, but not too, and we defreeze them, actually boil it till 50 degrees Celsius, so that all the enzymes could be activated. So sometimes whenever you're using FCS you have to boil them to 50% also, or 50 degrees Celsius. Then comes the gas phase. It should be full of oxygen. That should be around aerobic metabolism for atmospheric 20%, tissue levels between 1 to 7%, and carbon dioxide's buffering. Then there is a CO2 incubator. Um it should include controlled carbon dioxide, humidified 37 degrees Celsius should be used. So this is how your chambers where you put your cell culture for overnight to grow them, to give them the conditions for proper oxygen, carbon dioxides, proper humidity, and also temperature. So these things are quite important in this CO2 incubators. And surely whenever you're putting your cell culture, make sure that your flask is being cleaned 70% with 70% ethanol, and should not be contaminated with any false media. If it is so, if I put this as a default, if this one is default one, this one, all other cell culture will be having the default one also. If this one has a mycoplasma infection, so this will also will have, and what will happen, all the people who are working in the lab, everyone cell culture will be destroyed, and this person who did this will be the culprit, and which is actually not good, is not good for our work. So this is how the animal cell culture goes like. बच्चे for Dharampachavan, for plant cell culture, it's a plant tissue culture in the plant biotechnology. This is totally different. So with that, I was teaching plant biotechnology. So you can go to my YouTube channel. So I have given one 10 days lecture there. So you can have a view over that. Then comes your pH control. This is your physiological pH, is 7. pH is also affecting a lot. If it is acidic pH or basic pH, it will totally change your cell culture. So because this pH is affecting the cell metabolism, growth rate, protein synthesis, and availability of nutrients to it. And carbon dioxides act as a buffering agents in combination with sodium bicarbonate in the medium. Then temperature and humidity. So normal body temperature is maintained of 37 degrees Celsius, and humidity is maintained at saturating levels as evaporations can lead to changes in osmolarity on volume of media and additives. You can also have ingredients to so now let's check the contamination. So what are the sources of contaminations in our cell culture? So they are bacteria, yeah, fungi, mold, yeast, mycoplasma, other cell types, yeah, and free organisms, dust particles or aerosols, so surfaces or equipments. So this is how your cabinets looks like. So primary cell culture lines from so this is done to remove muscles from mice to protect the product only. So this is one way, and this is also class 2 cabinets. Here you have a glass over it. This is a laminar air flow. So all the air with the HEPA filter is going from inside and outside like this. So the air is not going, not able to go outside. So whatever the air is present, it keeps circulating within it, so that it can remain protected from any contaminations. And here you can see at the bottom these yellow plastics. So these are the yeah, there are three security levels, safety levels. Work has to be done. So all the cell culture, all the pipettes are being thrown here, and your lab coats being done there. So this is how you have to do your work. So this is the HEPA filters, laminar airflow is being shown to you. So you have exhaust fan, exhaust HEPA filter, laminar airflow, a laminar HEPA filter, and air being coming from outside and is being distributed from the back and then coming back from the top. So it's nothing like any bad air is coming. So there's an air barrier here. So it's not able to let in any bad air from outside. So human also are the reason for the contaminations, because we shed skin, bacteria, hairs. It could lead to some bad things to our cell culture. So to avoid this, there should be aseptic techniques, control environment of airflow, sterile media, avoid aerial contaminations of solutions, avoid manual contaminations of equipments, avoid repeated opening of bottles, keep cleaning with it 70% ethanol. And also you can use UV light before you start your experiment and also afterwards. So there is a UV lamp within your cell culture. So you can turn it on before doing experiments and turn it on after experiments. And only use disposable equipments, not you can reuse them again. So these are the various aseptic techniques. A scientist is being doing it with the lab coats wearing it, gloves, tips does not touch the tube also, holding the tube in the right way with the gloves and all these. So this is the correct way of doing your cell culture in aseptic manner. Then comes your pipette aid. So this is how you can take your media from your flask into your cell culture. Power of battery operated, motorized intake and expelling of fluids, transfer from one sterile container to another. So transfer a pipette, disposable enclosed plastics, package is sterile so that contained air is sterile in it. So you can also use these pipettes to have your media go in. So this is the size that matters a lot. So for ultra, I mean the poliovirus size of zero point 0.2 micro, then bacteria zero point 0.4, erythrocytes 10, smallest human diameter is around 10. So that's how to see that nothing goes bad into your medium. So we can use these very nice what is this thing called, membrane filters of 0.2 micrometer length, because from them when we take the media and everything will be filtered out with this size of membrane filter 0.2 micrometers. So in lab, this is how the skeletal muscle formation is being is done. That is your myogenesis. So myoblast proliferation in 0 hours. So in differentiations after 42 hours, the cell will be aligning. Then in 52 hours, they start to fuse. Your nucleus are coming together, and then 66 hours myotube maturation. So multinucleate myotubes starts to form. So it take around 66 hours to make your myoblast, your myoblast cells to be fully mature, and spontaneous contraction begins, and these cardiomyocytes will start to beat in your experiments. So, I can show you maybe. Oopsie, doopsie.
Your heart beats approximately 100,000 times a day to circulate 2,000 gallons of blood. The force exerted is similar to squeezing a tennis ball in your hand every second, 24 hours a day, for your entire life. The heart has tremendous endurance. Cardiac muscle cells are called cardiomyocytes. These cells contain specialized organelles such as myofibrils, mitochondria, the sarcoplasmic reticulum for storage and release of calcium, and the sarcolemma, which is a membrane that encloses each muscle cell. Cardiomyocytes are very flexible as they shorten and lengthen to perform the mechanical function of a beating heart. They are connected by cellular bridges, which allow the heart muscles to act as a single coordinated unit. Recent evidence suggests that cardiomyocytes are slowly regenerated and replaced with age, but more than half of the cardiomyocytes you are born with continue to function throughout your life.
No, I don't want to show you this actually. I want to show you how cardiomyocytes are beating. This one. So you can see your cells are beating. Can you see this? Maybe I can share you with the video, that will be more better. How cells are beating. You can check them later. So let's continue our cell culture thing. This is also muscle regeneration, likeness to formation of myotubes in culture. And this is how various cell cultures are being showed with inverted face microscopy. And this is how where the magic begins with the inverted microscope. So most of the cell culture at the cellular level, you can do plenty of experiments, many things within few months. You can publish 5, 10 papers if you want to, but to have a good journal, to have a good work, it takes years. Many PhD students, maybe one PhD student, and yeah, a good work, a good scientific work takes 3 years to publish one paper out of it, and build some good identity in the scientific society. So whatever the work that you will do in your future, whether it's a plant cell culture, animal cell culture, please do it ethically. Don't play any false thing on the long run. So in my PhD, I did one work with cell culture, and so far I'm able to publish one paper out of it in the in the Nature scientific reports. Yeah, but I'm very much happy that I was able to publish one paper, and nothing was false in that. Nothing hidden things were there. Everything that I did was with my honesty and truth, and that's what the you can die happy with that actually, because false things are not good for us. So then comes proliferation. So this is the proliferation after 24 hours, but the same proliferation with the fusion media after 7 days looks like this of your C2C 12 mouse skeletal muscle culture cell lines. Then you have to count your cells with the hematocytometer. So what you have to do is you count your cell in this chamber and this chamber, this chamber, this chamber, then divide them by 4, take the average, and that will be your number of cells in it. So just multiply with number of like dilution factor, how much media you have, then you can calculate an average cell number in your cell culture. So in the media formulations, proliferations, maintenance, so you can HAMS F10 nutrient mix, 20% FCS, 5 nanogram per ml your BFGF. And in differentiation fusion, you can use DMEM media with 2% horse serum, insulin, and linoleic acid. So for fusion, media change from nutrient rich to nutrient poor. So induce withdrawal from the cell cycle given the thin sausage they go for. So sometimes we also want to check, apoptosis, necrosis, cell death, so senescence. So we also have to withdraw the nutrients from the media, and this will lead the cell into the cell arrest, and cell will not grow. And then you can check that part as per the scientists are interested. So you can check anything what that's why in your class 11th or 12th you are taught with the basics of biology of meiosis, mitosis, how the cell organelles looks like, what is the function of each organelles, how they are working, so that you can do in the future if you want to be a good scientist, you can manipulate with them and find the answers of your hypothesis, like why one medium, one protein is working like that. On based on that, you can find an inhibitor to create one medicine, as a drug discovery. Because if you are able to understand all these parts with the trafficking, cell movement, cell signaling pathway, how they are working, only on the basis of that you can start working for the diseases. So that's why it's not like single thing is important. So everything is all interconnected with each other. Then you do the analysis. So this is showing you the analysis of adult mouse skeletal muscles. So this is a primary culture, also contains fibroblast with the fixed stain for desmin. This is the mouse skeletal muscle cell line H2KB in 35 millimeter dish, fixed and stained for desmin. This is stained with this blue color. Whenever you see, this is nucleus, and this is done with the help of DAPI or Hoechst dye. And the desmin is the green to see the myotubes. So your myotubes, your cell are being visible like this, and your cells being visible like that. So that this is not required. I think you will have now another class being started soon. So I didn't saw the time. So I will share with you now tissue culture. So students, if you have 5 minutes, I will just take you 5 minutes to show you how the passaging of cell culture is done. So your other class can wait for another 5 minutes. So I will just take you 5 minutes.
The growth of cells in culture follows a standard pattern. A lag after seeding is followed by a period of exponential growth called the log phase. Cells should be passaged when they cover the plate, or the cell density exceeds the capacity of the medium. Maintaining log phase growth will maximize the number of healthy cells for your experiment. Before retrieving your flasks from the incubator, sterilize the hood and have all required supplies at hand. Examine your culture carefully for signs of contamination or deterioration. Handle the cells with care during transport. For adherent cells, remove the spent medium from the flask using a sterile pipette. Rinse the cells with a balanced salt solution, such as DPPS. Make sure to use a salt solution without calcium and magnesium, as these may inhibit your cell dissociation reagent. After rinsing the cells, remove the salt solution. Each time you aspirate liquid off the cells, place the next solution on quickly. Add your cell dissociation reagent to remove cells from the plate. Use just enough solution to cover the cell sheet. Consider using a gentle dissociation reagent, such as Triple Express, to avoid damaging your cells during dissociation. Make sure that the solution completely covers the cells. You may want to tap gently on the plate to help the cells detach. Use a microscope to confirm the cells have released from the flask. They will start to appear round as they release from the substrate and will move or slide when the flask is tilted. Complete dissociation is necessary. Do not leave the dissociation reagent on too long, especially if you're using a reagent other than triple. You may choose to manually break up lingering clumps by repeatedly pipetting warmed medium over them. A single cell suspension is important to achieve an accurate cell count. If you are using trypsin, the collection medium will need serum, or you'll need to use a trypsin inhibitor to inactivate the trypsin. If you're using triple, inactivation is achieved by dilution alone. No serum is needed.
All is good so far, students? You are with me? Yeah, that's good.
Transfer the cells to a conical tube and centrifuge to remove any residual dissociation reagent. The speed and time of centrifugation will vary based on your cell type. After centrifugation, you should have a well-formed pellet. Remove the medium from the centrifuge tube with a pipette and discard the medium into a waste container. Try not to disturb the pellet. Resuspend the pellet with warm, complete growth medium. Gentle pipetting will disperse the cells to ensure a homogeneous solution of single cells. Remove a small sample for cell counting. Trypan blue is used when counting cells to indicate the ratio of live to dead cells. The stain turns dead cells blue, but healthy cells with an intact membrane will remain white or colorless. Using the microscope, count the total number of cells and the number of dead or blue cells. Based on your cell count, determine how much additional fresh medium to add for optimal seeding density. Add the required medium, mix the cells gently, and pipette the solution into fresh flasks. Cap the vented caps on your flask tightly. If the caps are not vented, cap loosely. This keeps contamination out while allowing adequate gas exchange. Use a north-south, east-west motion to evenly distribute the cells and transport the flasks to the incubator. When passaging suspension cells, you'll begin by removing a small sample from the cell culture flask for counting. You'll follow the same counting procedure for adherent cells using Trypan Blue and either a hemocytometer or the Countess automated cell counter. Based on your cell count, add additional fresh medium to the flasks. Stay within the minimum and maximum volumes. This is important to maintain optimal air exchange and shaking flow. You may need to split the culture into multiple flasks. Cap the flasks appropriately, depending upon whether they are vented or not, and return them to the incubator. Every 3 weeks, gently spin the cells, remove the medium, and replace with completely new medium. This eliminates cell debris and metabolic waste.
Okay, students, all good. We see us tomorrow then. Same time. I will be in touch with you then. So if you have any question, ask me through Telegram. I will share you this video also. So that was all about animal cell culture today. So give me positively your assignment tomorrow. So it was told that there will be off on Saturday and Sunday, but what I want is that we should not take off. So we will be taking class tomorrow at 7 p.m. And we continue. And we continue like this. So we see us tomorrow at 7 p.m. I will write this message for all of you also, so don't forget. Everyone okay, students? If you want to start tomorrow at 7, do you want off on the weekends or you want to continue? Just give me thumbs up if you want to continue. Then I will write a message. Okay, we will continue. Okay, bye bye, students. Take care. Join the next class. See you. Bye. Keep smiling. Tata. Bye bye. Hasta la villas. And what is that? Choose auf Wiedersehen in German. So I will switch it off now.